Excellent. Good afternoon, good morning, everyone. My name is Filippo Miglior. I am the president of the Genetic Commission of EAAP, and today the chair of the webinar, Livestock Breeding for Improved Sustainability and Animal Welfare. We have, I'm quite excited, we have a great lineup of speakers from Austria, UK, and Belgium. And hopefully this will be a very great discussion and very fruitful. This webinar is being organized in collaboration with Neogen. Neogen provides an extensive range of agrigenomic services and solutions for food and animal safety. They work alongside breed associations, genetic evaluation providers, companion animal breed registries, academic researchers, and directly with individual farmers and animal owners, offering affordable genotyping sequencing solutions for various species. I really would love to welcome everybody to this webinar, but also to our annual meeting, which will be in fabulous Firenze in the beginning of September. Our Genetic Commission has organized many sessions, 16 if I remember correctly, and with many different topics. And here I just put some top sessions that we feel are going to be quite successful in Florence. So really, please come over. It's going to be great, beautiful city. We were just there last week in preparation for the committee. The venue is fantastic. It's very close to the train station, so hope to see a lot of you there. Just some information for the audience. After each presentation, which will be around 20 minutes, there is going to be a 10 minutes Q&A, but this is of course flexible depending on the speaker. You can raise questions in the chat box, and I will read this question and the speaker will answer. The webinar will be recorded and further available on the EAAP YouTube channel. So please, we keep on organizing this webinar, which have been very successful on different disciplines. And so please check our social media accounts for upcoming news on the next webinars. And learn more about EAAP by using www.eaap.org. So with this, I invite the first speaker, who is Gábor Mészáros. Gábor is an associate professor of population genomics and animal breeding at BOKU University in Vienna, Austria. He's committed to teaching and researching animal breeding and genetics, especially related to genetics of diversity in all species. His favorites are small ruminants and dogs, though. Gábor is the coordinator of the European Master in Animal Biodiversity and Genomics, an international master science program. In his free time, he creates educational video content for his Genomic Bootcamp YouTube channel. Gábor, please go ahead.
Thank you very much, Filippo, for the introduction. Happy to be here. So the plan is, as you see, I'll talk about genomics of diversity and something about the present, and also then perhaps some future perspectives. Well, I'm from the BOKU University. Is a new name of the previously called the University of Natural Resources and Life Sciences Vienna. Now it's BOKU University, and also we changed the logo. For the presentation today, so what you to expect. So there will be some overall discussions around diversity with a focus on livestock. Also, I will give a mostly, like you will see, as a more like a review style of talk, so they're not very specifics or something very exact things, but yeah, just kind of reviewing what's happening, and well, perhaps providing some of my options and maybe some things on the future, and perhaps we can then discuss. So what is diversity or genetic diversity? That's our first kind of a basic question. I'd like to start from the basics. So you might see this triangle or this pyramid from time to time. So that is basically just saying that diversity itself is not a topic or a kind of issue that could be defined in a very strict way or defined in a very exact way and no other definition would apply. So here basically the diversity, depending on the angle you look at, could be just differences between the ecosystems or between the species within the ecosystems, or subpopulations within the species, and the subpopulations in livestock breeding or agriculture we call many times breeds. So this could be something along these lines, or it could be just the differences between the individuals within the breeds and also the underlying genetic characteristics, alleles, and so on. So it could be a wide range of things that how you could define genetic diversity or biodiversity. Of course, there is a substantial overlap between other life forms, I would say. So it's like wildlife or between wildlife and livestock, that would be more straightforward. But of course, the same things apply also for plants and the microbiome as well. Of course, all of these parts have their own flavor, but generally they are very similar in terms of what we can do and what we should do, honestly, in these parts. Well, if we say genomic diversity, so it's a new word in genomic, is so basically in this case we study the diversity based on the analysis of the DNA. Well, this could be based on multiple factors or levels. It could be a whole genome sequence data, the very popular single nucleotide polymorphisms of SNPs, or other methods. So the genomics itself is actually it's kind of a thing that is just a tool to determine the genetic diversity. So it's basically a possibility how to do better the things as we've perhaps done before, but it's not something that would be something absolutely different from the status quo. Well, I say here among others, because of course genomics can be used for a range of other purposes, not just the study of the diversity. So, but in general, what we have is we use genomics because it's useful and advantageous for us, and that is we improve the pedigree quality, or we can also replace the pedigrees, or basically if we do not have pedigrees, then we are good to go also without them for some of the characteristics. The genomics also provides more effective use of genetic variation and to deliver genetic gain. Also, we identify the deleterious alleles or understand the genetic architecture of diversity in traits, including those under selection. So here is a pretty nice picture. Well, I found it nice. It's from a paper, Determinants of Genetic Diversity, and you see here in the middle the genetic or genome-wide genetic diversity, just to add another phrase into the mix, but basically that means the same. And so there is a range of factors and kind of these valves that kind of influence the genetic diversity. So we have the effective population size, and these mating systems, recombination, selection, mutation, and well, there are a bunch of other things around it that is like if they increase or decrease, they might increase or decrease some of these elements that in turn increase or decrease the genetic diversity of a population. Well, but then I thought we can look at this picture from a bit different angle, and then it's like determinants of livestock genetic diversity, and just I will just try over this one. So what we have a few things that I would also mention in this presentation. So the effective population size is kind of a staple of these discussions. Now these mating systems, recombinations, they have to do something with the inbreeding or admixture. So depending, of course, where we want to go or what we do with our populations, but well, this is something that is influencing the diversity quite a bit. Of course, there is selection and also there is mutation, but mutation, honestly, of course, is present everywhere or in each species and breed, but is not really considered, I think, to a large degree in livestock, because well, just the generation intervals are kind of long, and there are not so many offspring. So I think the mutation effect of mutation might not be as pronounced in a classical sense. But nowadays, what we have is also, of course, the insects for food and feed. And yeah, so there's exactly the opposite. So of what I told before. So these very short generation intervals, very many offspring, many meiotic events. So yeah, I think also something, a mutation might be picking up also in importance when we are talking about actually the assessment of genetic diversity. And of course, well, there are also some other things here, but I move on. So how we can use the genetic diversity? Well, this is kind of my own categorization. So just maybe, well, just you see what I mean. It's basically, we can use the genetic diversity to make populations more diverse. So it's just kind of straightforward. We want to enhance the possibilities for adaptations. We can use also the diversity to make populations more uniform, and it might be a bit counterintuitive, but what I mean here, that we could use the kind of diversity measures to conserve the populations of breeds, so to actually protect the breed so that they remain in their purebred form. So in this setting, we are basically trying to make them more uniform and maybe avoiding the admixture situations. Or there could be various occasions when we use the mixture of the two approaches. As an example for the adaptive mechanism, so I kind of borrowed this example from wildlife, actually, just because, well, we have in livestock or in agriculture, so there is an element of the human element, and we try to protect the animals, but on the other hand, the nature is quite brutal. So in this case, what they kind of looked at are the effect of diversity on surviving translocations in Mojave Desert tortoises. So what we see here, that there is a significant difference between the group of animals. So basically, if they were less heterozygous or less diverse, they were more likely dead than the survivals. So basically the animals or the tortoises who were more diverse were more likely to survive. So it's basically just proving the point of the adaptation mechanisms, or kind of favoring the adaptations of the more diverse individuals. I'm sure there should be also similar cases also in livestock. For the adaptation / uniformity, so this is kind of, if we things go both ways, and we might think about the genomic inbreeding as you know, which goes into both ways. So if we want to well, we want to avoid, so we avoid genomic inbreeding if we want to increase adaptation, or we embrace inbreeding if we want to increase uniformity, in this case, that if we want to select the animals and then basically produce a stock that is better than their predecessors. So, well, probably it doesn't come as a surprise to you that genomic inbreeding can be determined in various ways, and the runs of homozygosity is one of those very often used methods that actually identifies the genomic regions that are coming from common ancestors, that are actually individuals that appear in family trees of both parents. And well, I take this picture from McQuillan et al. So this is, I think, one of the first, or if not the first, paper dealing with the runs of homozygosities, and just basically showing that this simple pedigree, basically on the both sides, these sections are coming down and are getting into a homozygous state in the offspring. And what we do in this case is we just calculate the so-called genomic inbreeding coefficient, which is nothing else than the sum of the length of the runs of the homozygosity divided by the length of the autosome. And in this case, what we do is the genomic inbreeding coefficient is the proportion of the genome in the ROH segments. So There is of course, the wild scale use for the context for the breeding, and there is also these average inbreeding levels provide very useful information. One other thing that I want to mention briefly is admixture. When there is one breed, there is another breed, and if they mate, they create something in between. Of course, we can look at the admixture levels as well. So the genomes of the two more previously separated populations are combining, and then we have we can actually show exactly the populations that are there. How we can use this information is, for example, if we want to ensure that there is a pure breeding happening. That was in one of our case in one of our projects in Burkina Faso. So that was in this group of farmers. They were very much interested in purebred animals, but as you see in their stock, there were quite a number of crossbreds as well. So here, just using the genomic information is crucial to identify which one of these hundreds of animals are useful for our purpose of really producing purebred animals. Effective population size is another one which is kind of mentioned quite often, and I would just touch this topic. So that is the census population size, that is the number of individuals, and actually the effective population size is the number of individuals from an idealized population that would manifest the same extent of genetic drift and inbreeding as the population in question. So basically, it's a kind of a smaller sized population that is somehow describing the variance within this population. Now, when we use genomic data, normally or usually we use either inbreeding levels or linked with disequilibrium to determine this effective population size. The values are then a bit debatable sometimes because again, there's a bit of this clash of the interpretation of the same thing between different fields. In wildlife, you could see this that the population should have more than 500 effective population size to thrive. Sometimes they go even like it should be more than 1,000. Well, what we would say in livestock, there's no way we have any of those values in most of the livestock populations. Yeah, there is the kind of a threshold value. It should not go below 50, but other than that, I don't think we have like those kind of hard high numbers. So okay, that's something to perhaps think about. And what my opinion is, honestly, is that effective population size, although used very often, it's more like an indicator rather than a strict criterion for the genetic diversity. Yeah, of course, there is a bunch of other things that we could talk about, but we don't, because the time is running quite quickly. So I was just having this kind of bit of an overview of possibilities. But then, well, I wanted to also talk a little bit about the, if not the future, but basically the current situation and what we could do perhaps. So what we have currently is genomics, let's say, based on the SNP data and partly on whole sequence is kind of a common practice with a bunch of methods. There is a really nice paper of harnessing genomic information in livestock, and so this is it says that there is this yeah, so it's completed this hype cycle that is kind of for the genomics part. They also say once in a lifetime, which I would debate very much, because there's, of course, there is gene editing, and now there's also the rise of the artificial intelligence and various applications. So I think we will see more of these hype cycles for better or worse in our in the coming years. I'm quite certain about it. Okay, just quickly what it is. So it's basically it's almost like a societal behavior, how people react to technology. So basically there is something new and we have overly high expectations. Then we find out that, well, expectations were indeed too high, and we have almost like a disillusionment process. But then hopefully the things work out for the better. And here we had exactly the same thing with the genomics, the emergence of genomics, and then 90s just saying QTLs, now we don't need anything else, and everything is solved. Well, found out quite quickly that is indeed not the case. But then there were some new possibilities of SNPs, and all the stuff with the sequencing that caused that what we have now is the common use of genomic methods. So this is like overall genomics, but as I say, for the genome editing could be something similar, or also maybe some other things that I even don't consider here. Current status of the use in Europe, quite okay. Well, we use genomics for pretty much everything. Production focus for larger breeds, and diversity focus normally for smaller breeds. Elsewhere, that's a question. Sometimes it's the same or similarly, depending on where you are. So it's like particular regions and institutions are doing good. Many places are not doing as good in terms of the use of the just so that they have the genomic data readily available for use in a wide scale. So the actually use of genomic data is more than certain, but I think what we have a big opportunity, and I personally think we should aim for, is to aim to genotype entire populations. So because we can then achieve a form of a big data. Well, big data will be also coming up later today, but so a form of the big data if you have a complete picture of a small population. So this, I think, is especially attractive for small breeds because, well, we can find what we have and what we don't have in our populations, in our breeds, and we can then act accordingly. So we have here the picture that you know very much, and this is the price of the human genome, and that was costing $100 million in 2001, and then went down to a kind of 1,000 or even below that in the recent years. Just a quick, so this is an immense decrease, and just to have a small kind of a side note here. So this is Lamborghini Diablo VT6 that was produced in the same time when the human draft genome was produced or drafted. A friendly price for this one is 350K. And if the car prices mirror the sequencing, then this car would cost 350, which is well, you could kind of see the difference there. That is immediately affordable for everyone. But the thing is that the genomics had this immense decrease, but it's a rate of decrease. It's not the price. The sequencing itself, although much more affordable than in 2001, it's for using for entire population is still a costly endeavor. Yeah, in particular, as I mentioned, some cases, some countries, institutions are much better off, some not so much. So for me, what I think what would be great for the future is cheaper methods of sequence like genotyping that would be very beneficial for the populations and to genotype entire populations. Why? Again, why entire populations? Why I insist on entire populations is that then we can truly find the rare variants that we don't even know about. And of course, we're finding these rare variants nowadays, what we have is genome editing. Well, again, probably this crowd, I don't have to introduce genome editing. This is just basically replacing the gene of our, so what we wish into the genome. And yeah, so again, genome editing, well, I don't want to now start the discussion if it's like GMO or not, but I'm quite certain that it is here to stay. And it will be utilized. Again, to which degree it will be utilized, I don't know, but certainly it will be utilized for the future. Well, it works great for the genes of the large effects. for genes that have a large effect, the benefits are not so straightforward for genes that have a smaller effect, for example, resistance or tolerance traits or other sort of even production traits. Of course, there are exceptions. For example, the sleep gene that for heat tolerance seems to be very promising. Yeah, so, and why is it important? So there is, in particular in small breeds where the analysis of diversity is very common, then what we have is kind of this marketing, almost kind of a marketing statement that is very important to have these breeds because they might have some trait or genomic variant that could be, you know, useful for the future, for the larger breeds, for the mankind as such. So we need to conserve those breeds. So what I think is that the future is now. So now we have the means of utilizing any and all beneficial variants from the small breeds. So that is also the reason why it's a good time to screen the populations and find out what we have and what the treasures we have in our small breeds. And so this makes then the unique traits of local breeds, small breeds, more valuable than other. Also, I want to just underline right away that, of course, the existence of unique variants is not the only reason for breed conservation. So I don't want to say that if by chance a breed doesn't have some extremely unique variant, so that is not useful anymore or should not be conserved anymore. So this is not what I'm saying. Just I want to say that if the breed has, then it's even more beneficial for that breed and in general for livestock breeding. For the so with the conclusions, I would like to just briefly summarize that well, the genomic data is indeed available for use. So there is an application. There are applications for genomic diversity and many other purposes. Well, limitations exist, to my opinion, to complete understanding of genetic variants in livestock populations. But once understood, the unique variants from small breeds or elsewhere could be utilized for the next leap in the livestock production, which would also, of course, mean the next leap in the possibilities of food production and then achieving the goals of the SDG 2, the Sustainable Development Goals 2 for the zero hunger. Well, if you want to kind of contact me in any way, then well, I work at the BOKU University in Vienna, so there is my email. I'm reasonably active on Twitter, and yeah, so if you are interested, the Genomics Bootcamp channel, of course, is always on YouTube. Yeah, this was it from my side, and well, I thank you for your attention, thank you for your time, and looking forward for the questions.
Thank you so much, Gábor. This was a very clear and overview of this topic, so I really appreciate it. I just first of all, I saw one question. Yes, this is recorded and it's going to be available on the EAAP YouTube channel. I don't see question for you. We have a few minutes, but I put down one simple question, I guess, from the end, about if there is any appetite, in your experience, of breeds fully genotyping or even fully sequence their own population in the, let's say, the livestock area, in the livestock. Is that already happening? Is there money for it? Is there desire to do it?
Yeah, I don't know about any breed that would go, you know, like genotyping the entire population. Certainly, there are. If we talk about the SNP genotyping, so there is many breeds who genotype, for example, over all male progeny and many of the female progeny. But this is like a SNP genotyping, which is useful, but it's more for I don't know, for a genetic genomic selection or something like that. So I think what would be kind of the ideas that I try to float here. It's basically are on the more on the sequencing side, or somehow to find out the actual variants, not necessarily whole genome sequencing, because then we are actually getting into a high budget values, that is our numbers, which is maybe not very sustainable. But yeah, something similar that would enable to get the variants from enter breeds, and then pinpoint the ones that are interesting. But for that one, it would be really useful if we could have like a lot cheaper sequence, like genotyping measures.
And of course, Gregor, Wim, you can jump in and ask question anytime. So from this last one, I mean, with genomics, we've probably seen the gap between the cosmopolitan breeds and the local breeds kind of becoming larger and larger. And so my question to you is, one of the problems is exactly that in a smaller population, we don't have enough numbers to provide gains, at least in the genomic prediction part, to have more success in genomic selection. So how do you, what is that kind of, not number, but the range of number which say, yeah, okay, you can do something on this population because you have enough data, you can find those SNPs that may be unique variants, so on, or we can just gain 5%, 5 points in reliability versus 30, 35 in Austin, something like that.
No, I mean, this is a bit of a difficult question, but my opinion on this perhaps is not so. I would just perhaps answer with another question, that why should we chase the very same thing with each population? So we know what is the best milk producing breed in the world. And also, well, the beef is a bit more maybe but more questionable, but there are also, I don't know, 5 contenders, let's say. The small breeds would never catch up to these breeds, never, with the productivity or so. So there you can go the way that many of them go. It's like, product quality, local products kind of type of thing that is like gets a unique value and the benefit. But then with this kind of setup, there is also the possibilities of just variants that are there that could be used worldwide, or also in these best breeds. And and then this is something that it makes them extremely valuable, even more if they would now just double the milk production or something like that. So I think that is something that we could think about. And yeah, so then I would not say that we now need. Of course, then there is that the question becomes difficult at the point when you say, oh, you don't need to do anything in these small breeds. That's also not what I'm saying. Just kind of do not have expectations that suddenly these breeds would be now top sellers for some reason. Yeah, so it's kind of this kind of finding this balance of the utilizations, and I think there is many uses also.
And it was a question without an answer myself. I was just a discussion. We have a question in the chat box. So this is from Anika. How do you think we can keep these small breeds alive for their diversity without getting too much inbreeding? You can stop sharing, and then you can see the chat.
Okay. How you can keep the small breeds alive for their diversity without getting too much inbreeding? Well, okay. So, I mean, getting some inbreeding is like unavoidable. So I think what we should do is not to get like an increase of inbreeding between the generations or between years, so not as much as. So that is, and then it just, I think, comes down to planning of matings and basically animals that are not related with each other. So animals can be inbred themselves, but if there's two animals are not related, their offspring is suddenly not inbred or not as much. So it comes down to planning for breeds.
And then the second question, if you can read it.
Yeah, if local breeds are to be used because the unique trait, e.g. high resistance, what would it be, the time horizon for that? Well, it could be even now. So the slick gene that I was mentioning, it is being utilized as we speak. So there is that was identified, and I know that there are efforts to put them in, or put this particular gene to high producing genes or high producing breeds, and make them suddenly much more viable for tropical environments where the heat is, of course, an issue. So if there is a trait, or especially nowadays we have a bit of a limitation that it should be as for now a gene of large effect. So that is I think that is what we have to acknowledge. But if we have something like that, it's immediate. It's, of course, it's a function of money, because there is some research and all that stuff needs to be done. But other than that, it's pretty much immediate. And I think with the kind of development of these technologies and all this kind of ever increasing pace of this development here, and also some other parts of the livestock industry, I think this will just become better and better in terms of applicability, and we are kind of possibilities to finding these genes and then applying them into the proper populations that then really can make a difference.
Thank you so much, Gábor. I appreciate. And I think we move to the next speaker at this point. And I would like to introduce Gregor Gorjanc. Gregor leads the Highlander lab at the Roslin Institute in Edinburgh, Scotland. He and his team research management improvement of population using data science, genetics, and breeding. And they're particularly interested in methods for genetics and breeding, design and optimization of breeding programs, and analysis of data to unravel biology and to find new ways of improving population. And today's presentation will be about storing and analyzing mega-scale genomic dataset. Gregor, go ahead, please.
Filippo, thank you very much for this nice introduction. Let me just share my screen. Can you please confirm that you're seeing my screen?
Yeah.
Good. Thank you very much. And thanks also to the organizers of this webinar, the EAAP group, as well as the sponsors, Neogen, for getting us all together here. I think this is a really good opportunity. So I'll jump straight into my talk, and I really like to also thank Gábor, because he kind of laid out sort of what has happened in the genomic space. And I kind of started my training and career just before genomics really became very widely used in livestock. I've done all my training before of that, but obviously then, when genomic data became widely available, I've started to use genomic data, mostly for genomic prediction and genomic selection, and kind of in the space of quantitative genetics. And it was very exciting last 15 years. I still remember when I saw first papers published in around, from 2001 from Moivizen et al., up to 2010, when we saw a couple of publications with something like 1,000 individuals that were both genotyped and phenotyped. But before we figured out some properties of this data and what we can do with the data, within a couple of years, we already had reports of data sets with something like hundreds of thousands of individuals that were genotyped. And today, we already have some populations where a lot, if not all, of individuals are genotyped. One example is very close here is in Ireland, where I believe they're genotyping pretty much all of born beef calves. So they are already with data sets somewhere around 1 million, and this is not the only case. And if we just extrapolate what has happened in the last 10 years and we try to project that into the future, it suggests that in 10 years we will have populations with billions of individuals genotyped, which obviously sounds completely ludicrous. But if we turn back time for 10 years, and if you would, we would predict that you know, in 2010, that we will have millions of individuals genotyped, nobody would believe that either. But, you know, we are here today. So let's see what the future brings. Now, all of this has been done with SNP array genotypes, but on the back of that, you know, very exciting time and pace, we had another wave of genomic revolution, which was, you know, introduction of whole genome sequencing, and that's the graph that Gábor has showed. You know, the price of sequencing, it really has dropped, and it's continuing to drop. And in, you know, around 2015, we had data sets with about 1,000 individuals where we had whole genome sequence. The 1,000 Bull Genomes project is one example from our community, but there are other such similar projects. And today, we already have data sets with something like 10 to potentially up to 1 million of individuals that have whole genome sequencing data. In some cases, this is done with sequencing and imputation, but in some cases, it's done just with sequencing. For example, here in the UK, we have a project in human space called UK Biobank, where 500,000 people have been whole genome sequenced. So you see, we already are in the space of having potentially up to 1 million of individual sequence, and there are other projects, one in US that I believe is even bigger. That's in the human space, but you know, in agriculture, we're not lagging behind too much. So again, what will happen in the next 10 years? We will see data sets with millions, potentially 10, 100, maybe even billion of individuals with whole genome sequence, which I know sounds really crazy, but if you look at what has happened in the last 10 years, you know, it's probably what's going to happen in the next 10 years too. But now we have like a huge problem, and this is where the mega word from the title comes in. We now have these data that essentially is stored in some sort of a rectangle. Right here, I'm showing my samples, and I'm just assuming they're haploid in columns, and I'm showing variants in rows, and you know, columns times rows gives me this rectangle. And the complexity of storing this data is by definition sort of quadratic, right? I need to store n columns times m rows. So that means I have quadratic complexity on storing this data. And just in terms of, you know, the data set sizes that we were discussing in the previous slide, where we were talking about potentially 1 million of columns and, you know, many millions of rows, if we're talking about whole genome sequencing data, these data storage will just not scale. And a couple of years ago, I've came across this paper from a collaborator down in Oxford. His name is Jerome Kelleher, where his idea was that this data is just an outcome of a particular process. So instead of working with the data, we should actually work with the process, because that will give us some computational advantages. And this genomic data is data about DNA, and DNA comes from the process of DNA inheritance between generations. And in many spaces in biology, they are using this type of data structure. And the idea here is, if you think about having an individual back in past, this is a string of DNA. Let's call it I, has at some point duplicated, and then we get a left branch and a right branch, and then you see that on this right branch we got a piece of DNA that we call G, and then that G then duplicated again to get DNE, and something similar happened on the left-hand side, right? So this is just the simple act of DNA copying itself. But as we know, this DNA copying is not perfect. Every once in a while, we do get a mutation, and these are these red squares here, diamonds, that are denoting these mutation events. And these mutation events are then generating variation that we see today in our samples. Now, the beauty of thinking about this tree and the process is that we can encode the process very efficiently. Specifically, I can encode the shape of a tree with this tree table. For example, I can write that haplotype A, this piece of DNA A, came from H, which is denoting that we have this link, this edge or this branch between A and H. And this table tree only has 2 columns and a large number of rows, but the point is that the table only grows in the number of rows. Then we have the variance table, which has only 2 columns, and it's denoting where in the position we saw mutation and above which node the mutation has happened. So for example, this mutation 7 at site 7 has happened above haplotype G, which means that both haplotype G, D, and E will have that mutation. And if that mutation is a very successful mutation and it's kind of spread throughout the population and is now in very high frequency, we only need to stare that mutation event once, which will give us a lot of advantages in storing this kind of data. And this type of tree thinking is actually really common to much of biology. And I think in our community, we are not tapping into that knowledge enough. Many of you will be familiar with the book of, On the Origin of Species from Charles Darwin. And that's, that kind of started this idea of tree thinking. This is, sort of the first phylogeny that was ever sort of, scribbled down on a piece of paper. And you can see that Charles has written, I think, he was not really sure what was going on. And just when I was preparing this set of slides, I saw on Twitter this very nice post here by Leo. And Leo here very nicely shows that, if I start with these black circles, imagine them as being genomes of an individual, then you will see here that, first and the second genomes, they represent an individual. Maybe think about that, that's me. And then obviously, going further back in past, I am somehow connected to all other individuals here both in the panel and among the audience, because we're all members of one species, which is the human. But of course, the way we group ourselves is quite complicated. But this has a very natural sort of hierarchical structure in terms of how populations have evolved over time. But I would like to emphasize that when you will be looking at this literature, it took me a while to sort of figure out these terms. Colleagues use the word tree for many different things. So if you're talking about a relationship between species, then you will be talking about species tree. And maybe some of you might have looked into human genetics, for example, and then you will see that chimps and bonobos are more closely related than we are to chimps. But then we are more closely related to these two species of apes than with gorilla, right? But that's sort of at the species level. Then quite often what, in our community in animal breeding, we think about different populations of breeds. Being based here at Scotland, which is sort of the home place of Angus breed and Ayrshire breed, they probably just, at least by location, will share a common ancestor. But then Holstein is another taurine European breed of cattle that will be more closely related to these two than an example of a breed from an Indicus family. Of course, quite often we actually don't want to work just at the population level. We want to go at the individual level. So that's me and my sister. We're probably the most closely related. Among these four people listed here. But then Gábor sort of has a bit of a similar Slavic origin than me and my sister have. So probably I will be more closely related to Gábor than to the third speaker today, which is Wim. But that's at individual level. We might go then another level down, which is the gene or the locus level. And this is then where we're really looking at the ancestry of different alleles at the DNA position. But what I've only shown now is copying of DNA, and then mutations happen. But we know, as Gábor has pointed in the previous presentation, is that the majority of variation that we kind of work with on a short-term basis actually comes from recombination. And this is, here I'm showing on the bottom left, I'm showing an example where we're crossing three parents. We're creating two crosses. You see this wide variety of difference in the progeny, which is in this particular case just due to recombination. I didn't introduce any mutation here. And on the right-hand side, I'm sort of showing the same thing. I'm kind of pulling two full SIB individuals from, let's say, this wide distribution. This white circle is, one individual, and the other white circle is the other individual. They are two full SIBs. They both get half of DNA from mother. This is the red piece. They get half of the DNA from father. This is the blue piece. And then when we go further back in time, we are inheriting DNA from our ancestors. However, because of the process of recombination that happens during meiosis, we are in fact inheriting different bits of our ancestry, by inheriting different pieces of DNA from our parents. So how can we take this tree thinking and kind of infuse it with recombination? And we can address this by a technique that's called ancestral recombination graph, or ARG for short. What is ARG? In very simple terms, ARG is a collection of DNA trees along the genome, and essentially describes all the events that have generated the DNA of our samples that we're working with. So we need to store essentially three events. We need to store this branching, when we have a piece of DNA that copies, right, it branches, or if I think back in time, I have two pieces of DNA and back in time they coalesce into a common ancestor, then I need to overlay mutations on these branches, and then I need to be able to introduce both recombination and gene conversion that changes these type of trees as we go along the genome. And this is really the ultimate population genetics object, right? So we really want to work with ARGs. However, until recently, they were really just more or less theoretical, interesting thing, but they were really impractical to work with. But now, these days, ARGs are becoming possible to work with on a large scale. Now, for you to understand really what these ARGs are and how we use them, I'll show you this example and walk you through. So imagine here we have a pedigree. We have two parents that they have been crossed to create a progeny, and then the progeny has been back crossed to the left parent to get another progeny, right? Sort of a grandchild here. And I've painted the chromosome with respect to the parents so that you can track the chromosomes. So you can see that this violet chromosome has been transmitted without recombination, but this first child got a recombination between the green and the orange chromosome of the right parent, and then when we did the back crossing, we got the full blue chromosome, and then this recombined chromosome has, the green and orange recombined to the violet, and this is the chromosome that I got. In this particular example, we have two recombinations, which means that we will have to look at DNA tree at 3 positions. The first position will be before the first recombination, second position will be between first and second recombination, and third region will be after the second recombination. So if I now take this pedigree and I slice it according to these 3 regions, where here at the bottom you see I have the first region, then in white I have the second region, and on the right I have the third region, I now have these pieces of DNA, and each of these trees here that I am representing is a tree representing the ancestry of each piece of DNA in my sample, and this is something that's in the population genetics community called a local tree, because it's tree for this local piece of DNA. Now, one thing you can notice is that in parents, essentially nothing really changed, right? You will remember that this N8 chromosome was pink, and of course it is now pink or violet everywhere. And what you will start to notice is that these local trees only really change when we have a recombination. So you will recall that the first child got a green and then an orange piece. And you see this is this transition here, but otherwise there is no change for that particular chromosome, neither is for the other chromosome of that individual. And again, we have another change. When I look at the grandchild, we have the green, then the orange, and then the violet piece. So while this is very intuitive representation, it's not very efficient representation because we kind of keep repeating redundant information. And this is where the idea of the collaborator down in Oxford kind of came about, and that was, let's try to encode only bits that change when I go from left to right, and from past into the future. And this is where a concept of a tree sequence has been born. The idea is that we will try to represent this sequence of local trees with another sort of a data format. This is what is usually called a tree sequence in that community. And tree sequence is just a format how to encode ancestral recombination graph, which is all about encoding the events that has happened around the DNA. So what I'm doing here now is first I will introduce a table that is called a node table. I'm listing the names of these chromosomes that we call in this community node. I am also writing down the time when those existed, if that time is known. Then I'll introduce another table called edge table. This is the table here on the right. And the purpose of this table will be to encode relationships between haplotypes. And let's start with 2 very simple relationships, and that is that this blue chromosome N7 has been passed to chromosome N2 without recombination. So this creates an edge or a branch, connection between them. And I will encode this in this table, saying that edge 1 is representing passage of DNA from the ancestor N7 to the descendant N2. And this is a whole chromosome, which I'm here denoting by centimorgans, from 0 to 100 centimorgans, assuming that the chromosome is long 1 morgan. I have the same kind of a situation for the H6, that's denoting relationship between the chromosome N8 and N4. Of course, the interesting thing happens when I have recombination, and this is the example of a recombination between my node, for example, here N1 and N3 and N4, where I will have two edges. Namely, this descendant N1 got a piece of DNA from chromosome N3 and piece of DNA from chromosome N4, and then I just need to denote where those recombinations happened. You will recall from the example before that this first piece of DNA came from N3 from 0 to something like, 3/4, or you see 70 centimorgans, and then the other piece came from from N4. Finally, I have another recombination from the right parent to the child, and because of one recombination, I get then two edge records. Okay, this has now resolved all the recombinations within this pedigree, but then we can actually go beyond the pedigree back in the past, and this is where the concept of coalescence and coalescent, which is a data structure to describe the coalescence events. And what I'm saying here is that this piece of DNA in N7 and N8, they have a common ancestor in N9. This piece of DNA in N5 and N6 have a common ancestor in N10, and N10 and N9 have a common ancestor in N11. What you will notice is that when we go back in time, these pieces of DNA are getting shorter and shorter because there were more recombination events that were happening that were kind of chiseling down these segments. And of course, then in the next, sort of in the next region of the DNA, the ancestry will change somewhat. And then our task again is to just encode these edge events like I've done it before. So this has now captured all of the branching/coalescence events and recombination events, and now we need to handle also mutations. The way we handle this is by introducing two new tables. I will introduce a table called site table that will tell me which are the segregating sites in our data set. I'm just keeping things very simple. Let's imagine we only have two polymorphic sites, site 1 and site 2. They might have a specific physical position in the genome. And what we need to know is what is the ancestral state at those positions. And let's say that someone tells us that this is a CT. So this root here will have, DNA sequence CT. But then, of course, when this piece of DNA is transmitted to next generations, we will start to see mutations. And this is what you see here on the left branch. On the passage between N11 to N9, there was a mutation where C mutated to an A. This is first mutation in our data set, so I'll store it in the mutation table. I need to locate where that mutation happened. It happened at site 1, and it happened above node 9, where C mutated to an A. Okay? Then this piece of DNA has been passed to chromosome 8 and 7 without mutation. And then on the passage between the chromosome 7 to 2, we had a second mutation in our data set where T mutated to a G. So that's a second mutation. It happened at site 2. It happened above node 2. And T mutated to a G. Okay, that's all fine. We've covered the left branch. Now let's go to the right branch. This segment N11 has been passed to 10, 6, and 5 without mutation. But then on the passage between 6 and 3, T has mutated to an A. That's a third mutation in our data set. So I have mutation number 3 at site 2. It happened above node 3, where T mutated to an A. And if you have been carefully following, you will notice that this is now second mutation that we saw at this site 2. We saw previously that site 2 mutated in the left branch. And what you will now see is that we now have different kind of a mutation. So ancestrally, we had a T allele. In the left branch, we got a G allele, but here in the right branch, we got an A allele, and this is how you see you can get multi-allelic SNPs or even some other type of polymorphism. Finally, we had a mutation number 4, where the chromosome 3 has been passed to 1, where C mutated to A. Fourth mutation at site 1, it happened above node 1, C mutated to A, and I've just made it so that here you see I'm repeating the same kind of mutation that we've seen in some other passage of DNA between generations. And of course, now the question is that mutation will have the same effect or not? But moving on with the computational side of things, I would like to show you two interesting graphs in terms of showing how this type of data storage can help in storing data more compactly, but also computing things much, much faster on this data. So on this plot here on the left, I'm showing the file size on the y-axis on log scale and the number of chromosomes on the x scale. And I would like you to focus on this intermediate line here at 1 million, 1 million is important in the sense that in UK we have that project I mentioned, UK Biobank. 500,000 have been whole genome sequenced. People are diploid, which means we need to deal with 1 million chromosomes. And if we would store that as a VCF file format, we need something like 1 terabyte of space. We can compress it and bring it down to something like 100 gigabytes. But when you need to analyze the data, you kind of need to decompress it. So more or less you need a terabyte, which in terms of storing on a disk is not a problem, but actually analyzing it and reading it into memory becomes quite a bit of an issue. If we would be able to encode this data as an ARG in this 3-sequence format, we would need something like 100 megabytes, right? So this is multiple orders of magnitude smaller data set, and then we can analyze 100 megabytes, pretty much today on every laptop. And importantly, when we will start to scale these genomic data sets, this is a way to scale genomic data sets, for quite a while. Similar advantages can be expected on the compute side of things. So instead of doing calculations on the rectangular matrices, we can do calculations on these ancestral recombination graphs. It does require special algorithms, but once you have those algorithms initially take more time for small data sets. But when data sets start to scale, those specialized algorithms that work on ARGs versus the VCF file or a genotype matrix, they scale much better. So let me show you a couple of results from a PhD student in our lab, Gabriela Mafra Fortuna. Gabriela is from Brazil, and she's very interested in working with both the taurine and indocin type cattle, particularly when they're crossed. An interesting part here is that Bos taurus and Bos indicus, there are 2 subspecies of cattle, and they have diverged since many years ago, something like 100,000 years ago, and that's why they are quite different, but not just because of drift, they're also different because they have evolved and been bred in different environments. So to kind of start this type of analysis, Gabriela has taken 1,000 bull genomes data that's publicly available. There's almost 3,000 whole genome sequences available from almost 160 groups defined by breed, crossbreed, etc. There's lots of diversity in this data set. And she's taken 29 autosomal chromosomes with more than 100 million variants in that data. She has then phased the data to get the haplotypes. She used ShapeIt for that. Then she inferred 3 sequences using a tool called TS-infer, and then she's analyzing these 3 sequences using a Python package called TS-kit. So to give you a sense of the scale of the data, when she downloaded VCF files, there were around 800 gigabytes of disk space. When she inferred the 3 sequence, the 3 sequence is on the order of 50 gigabytes, which gives us a compression of more than 90%. And then, running analysis on these ancestral recombination graphs becomes really fast. Here is just one quick summary of the data. We're sort of showing kind of a heat map, sort of a structure-like plot in this data set. And what you will notice here, we have a big block of animals, which are, these samples have been taken mostly from Holsteins. They are by far the biggest contributor to this data set, but you clearly see some other breeds that are very clearly represented in this data set, and it's, a lot of breeds, but I will not go into details. One interesting thing that we have done in this data set is that some of the animals that cluster with the Holsteins do not have breed known. So we have then looked at who are their nearest neighbors. That is, in this tree data structure, which individual is closest to that. If I take a focal individual, I do not know the breed of that individual. I look who are the closest genealogical nearest neighbors, who are the samples that are closest in this ancestral recombination graph. And then if we look at the breed of those individuals, what you will see by far and large is, here in red, they are actually either crosses with Holstein or with the brown Swiss, but there are also contributions from some other individuals. And something to be very mindful of when we are classifying individuals into different groups, which is what we're doing when we're doing breeds, is that is by definition sort of a wrong way to go about it. I know that operationally we would like to categorize things, but really we should be thinking about if I have one individual and another individual, instead of trying to sort of put them together into a group or put them further apart, is to think about how did those individuals came about, what is their ancestry, And that is a much, much more natural and logical way to go about. And really, we should be talking about, about different breeds or different nationalities almost, is more talking about is what is the history of, where we or sort of our livestock came from. Now, this is all, about managing data, doing some sort of population genetics on that data. But what is now really exciting is, can we use this data structure to do quantitative genetics on it? There was a very nice perspective piece published by Kelly Harris. And the idea here is very shortly, on the right-hand side, you see you have a group of individuals. Some are short, some are tall, and then again, some are short. And the reason that these individuals are tall is because at some point there was a mutation that has been passed down to that group of individuals. Now imagine that we did not type that individual, we cannot type that mutation. But if we have enough genotype SNPs around that mutation and we can reconstruct these genealogies or ancestral recombination graphs, we should be able to do this kind of quantitative genetics with ancestral recombination graphs. A couple of years ago, we published a paper with Maria Liesele. Maria visited us from Norway. We had a very nice collaboration. We've started doing this kind of models. You will recognize this tree from previous slides in my presentation. And we've developed quantitative genetic model that works on this type of data structure. In this particular case, this is a tree, but you can in a way think it just like it is a pedigree for these pieces of DNA, but obviously now here there is no recombination, we only have potential mutation. And then we apply these data in collaboration with colleagues from Croatia on some mitochondrial DNA. Mitochondrial DNA does not recombine, and we were able to estimate the effect of this tree. Colleagues have given us this tree data structure, and then we have estimated the effect of each of these pieces of DNA, but shared information between the pieces of DNA. What you can see here in red colors that we have pieces of DNA that seem to be decreasing the phenotype, while here, in the upper part of the tree, we have blue pieces of DNA that are increasing the phenotype. Now, this is for a single gene tree without recombination. And I'm sort of happy to let you know that last week I was down in Oxford. We had like sort of an international meeting. People from all different parts of the planet came together. We had like a lot of mathematical brainstorming, and I think we've cracked it. So please stay tuned. There might be some exciting things coming down the line. And last, before I close, I would like to let you know that we have an online course that's freely available. It's available on a platform called edX, and the course is all about breeding program modeling with an R package called AlphaSimR. And with this, I will stop and happy to take any questions. Thank you.
Thank you so much, Gregor. Appreciate. Are there any questions for Gregor? This was a tough talk, my friend. So I'm gonna struggle to find questions for this one, but I'm trying. Okay, so please forgive me. Can this approach be used not just for research, but also for, let's say, genomic prediction instead of trying to decrease the space that we need that is becoming more and more problematic, of course.
Correct. So I think the essence, and it took me a long while to actually understand this, so I appreciate it looks a bit alien to our community, but there are other communities, particularly across much of biology, where tree thinking is very natural to them. And it is very natural because this is what DNA is doing, right? And we really should be thinking about what DNA is doing as a sort of as a molecule. And it's essentially, it's just copying itself. Copying is imperfect. That's how we get mutations. And during meiosis, we also get recombination. And it's pretty much those three events that are generating the diversity we see among ourselves, obviously plus selection that drives different groups in different environments in sort of different phenotypes. So I think fundamentally this is the right thing to do because it's the right thing to do sort of from the event perspective. It's also the right way to store the data because that is what is generating the variation. And now the question is, can we do analysis on this data? It seems it will be possible. There are many algorithms that already work on this, like calculating allele frequency very effectively, calculating FST, various measures of relatedness or inbreeding, but not yet what I personally am very much interested, and I know yourself as well, is can we do genomic prediction with that type of data structure? And if we can, I think there is promise here that we will be able to sort of share the data between the big cosmopolitan breeds and the local breeds, where the data will not be shared directly, but will be shared essentially, via ancestors. Now, if ancestors are like really far back in time, probably there's a lot of data from something like Holsteins will not be very, very useful for a breed that's really, really, distantly related. But if there is a breed that's closely related that has a recent ancestor, there might be just a few mutations that separates them that we might be able to share the data more appropriately. But how will this pan out is really an empirical sort of research question.
Sounds like a very nice research project on its own, that I would say. Very, very promising. Gábor, did you have a question? Originally saw your...
Yeah, just a quick, it just came to my mind. So this type of approach is mostly foreseen for kind of a sequence-related variants, or I mean SNPs, or also some other type of variants.
So the approach is universal in a sense. We can work with any type of genomic data. So we can work with SNP array genotypes or whole genome sequence or some other intermediate form. So in that sense, there is no limitations, apart from if the genomic data has not enough markers, then the ancestral recombination graphs that we will infer will be very coarse. And then the question is at which point they will be so coarse they will not be informative. That I don't think we have an answer to that question in the literature yet. I've seen some application from human genetics where they are claiming that with a SNP array chip, they can infer ARGs that are pretty much of the same quality as with whole genome sequence data. So I think that sounds quite promising, but it's very early days. So I would, we need to study this closely.
The SNP array chip of which density? Because that is also could be a...
And that will again matter from a population to population. This particular study was in human genetics, where most of the time when they work with the SNP array is about 1 million, give or take, markers, but then genetic diversity in humans is larger than in livestock. So maybe our 50,000 marker SNP arrays might do just fine, but we need to study this.
Yeah. Thanks.
Thank you so much, guys. And so we can go to the next speaker. Thank you, Gregor, I really appreciate. Before going to Wim, I just want to mention again, this recording will be available. Normally, the recording for EAAP webinar is only available for EAP members, but thanks to our sponsor today, Neogen, we will be able to have this available for everybody on our YouTube channel. So now I can introduce Wim Gorssen. He graduated in 2017 with a master's degree in bioscience engineering, specializing in animal production. Following his education, he started working as a PhD student in the Center for Animal Breeding and Genetics at KU Leuven in Belgium. Wim focuses on quantitative genetic research in pigs, with special emphasis on selection for increased uniformity and resilience. Please, Wim, go ahead.
Hi, thanks for the nice introduction. You can all see my screen. Nice, I hope. Yeah, perfect. So as you introduced, my talk will be a little bit more practical. And it will focus on the new phenotypes to breed for resilience in pigs. And the results I will present mainly stem from my PhD project, which I recently more or less finished, also called New Phenotypes to Breed Resilient Pigs. So I will go over some examples of studies I did on new resilience phenotypes and how we could apply them in practical pig breeding. First, maybe a bit background about why we would need more resilient animals and more resilient pigs in particular. In pigs, the main driving force, in my opinion, is that we are having an increased number of piglets per farm employee. So about 40 years ago, an average farm had about 150 sows with 20 piglets weaned per sow per year for 1.5 full-time equivalents. So that leads to, maybe I can also point a bit here. So that leads to about 2,000 piglets per full-time equivalent. So, but it's now due to increased productivity of sows combined with intensification of the pig sector. This led that the number of piglets per full-time equivalent has increased with a factor of 10 almost, to about 20,000 piglets. So pig farmers, they really desire pigs which are labor efficient, which can fend for themselves and don't need care every now and then. More or less, additionally, animal welfare is getting more important. That's evident everywhere in Europe, I think. And also we need pigs that require a less amount of antibiotics. And as we want to keep them in more naturalistic settings, so outside, for example, we need pigs that can withstand more environmental variability, which are better able to cope with heat stress or cold stress, for example. So these forces are driving us to breed for more resilient pigs. And as Peter Knapp stated nicely in an article a few years ago, these robustness traits, these resilience traits, they were referred to as secondary traits like a few decades ago, but actually now they have evolved to the most important trait in animal breeding, maybe. So this is also evident in breeding goals. So this is a nice overview of EAAP on how breeding goals have shifted in time. So about 40 years ago, in the 70s, the 80s, we just came out of a world war. We were mainly focusing on increasing productivity, increasing efficiency. We needed more meat to feed people. But today, the focus has shifted more towards disease resistance and robustness, closely linked to resilience, also survival, also closely linked to resilience, and other traits like reducing emissions, carbon footprint, diversity, stuff like that. So we have a more diversified breeding goals with a strong focus on resilience. Now, we know we want to have more resilient animals, but it's resilience is a trait that is fairly hard to quantify. But we have a general resilience hypothesis that states that animals have an optimal theoretical production level. So when everything is fine, I used to say when the sun is shining and everything is nice, you have an optimal performance. But when you face a challenge, an environmental challenge, could be heat stress, could be a disease, could be aggression from pen mates, you will probably deviate from your optimal. And then you will have differences between animals where you have more resilient or less resilient animals, where more resilient animals will have a less severe deviation with a quicker recovery after the challenge. And this deviation is something we can quantify, and thus we hope that by quantifying this, we can phenotype it and breed for more resilient animals. So this is a theoretical framework to make it a bit more practical, concrete. I have a case example. So during my PhD, I have not only doing my research, but I will also be busy with a small scale experiment. So I've bred 2 little piglets. First we have anemone, and next we have hazel. So these are full sips. They are both genotyped and they had parents with good breeding value. So these piglets had they were quite similar, but they had a remarkable difference after birth. So after birth, it's normal that babies lose weight and then recover, because like birth is an extreme challenge, both on the mother as on the infant, of course. But we saw that anemone had a severe dip and a very slow recovery after the birth challenge, whereas hazel had her dip, but really recovered quickly and she grew a lot in a very short time. So what was the main difference between my two little piglets here is that my oldest daughter, Anemone, she's diagnosed with CFTR, so cystic fibrosis. So she has one recessive gene, making her more susceptible to diseases and making her digestive system more vulnerable. So one simple gene created these big differences between my two kids here. So this indicates that genetic differences can have an impact on resilience, and that we can measure this by looking at weight deviations in time. And just to say, Anemone got medications after 4 weeks old. She was diagnosed then, so everything was fine, and she's still very healthy at the moment. So this is the main background. I'm now going to try to show some examples of my own research on resilience in pigs. And I'm going to start real simple because we're developing a lot of resilience traits, which are quite complex, working with large data sets. Actually, a lot can also be done using fairly simple phenotypes. So I'm first gonna show something on a study we did in a Pietrais sire breeding program here in Flanders, in Belgium, where we used phenotypes on crossbred piglets, vitality, and congenital defects. So the data we had, it was from the progeny testing farm from the Flemish Pietrais breeding organization, where they recorded traits in the farrowing house at the litter level. So these traits are fairly simple. It's just per litter. Piglets are given a score on their vitality and uniformity. So vitality is scored from low 1 to high 5. And uniformity was scored as the number of small and large piglets. Next to this, we had phenotypes on the reproductive capacity and mortality. So the number of piglets born alive and dead, and mortality before weaning. And we had data on congenital defects, so splay legs and scrotal hernias, for example. So we had data for the Flemish Pietrais breeding organization, but in Flanders we have a quite unique situation where we also have an independent breeding program where every breeding program can send in semen of boars where they can be independently validated for the Flemish pig breeders. So we had 2 independent data sets with 384 boars appearing in both data sets. So we had quite some nice data, about 14,000 liters from the Flemish breeders, about 4,000 liters for the independent testing. So what we wanted to study now, the Pietrais sire breeding program has started selecting on these traits. So a combination of vitality, scrotal hernia, splay legs, and mortality before weaning since 2019, so about 5 years ago. So we wanted to study what is the genetics of these traits? Have we been making progress? And can we validate our breeding values between those 2 independent data sets? So first, a bit on the genetics. So these traits from crossbred litters, so they have a Pietrais sire and a hybrid dam. They're mostly dictated by this maternal genetics, the sow genetics. However, we do see here, we use the sire dam model, and on the sire side, there was still a significant heritability. So the sires still play a role. And especially for traits like vitality, scrotal hernia, splay leg, and mortality, the sire still explained quite some variance. So like if you have the sire dam variance, the sire explained 16 to 13% of that. So the sow was responsible for most variation, but the sire also had quite a substantial share in this. And if we then looked at the progress we've made since actively selecting this, so after introducing this in our breeding program, we have seen a reduction in congenital disorders. So like from 2020 to now, like a few months ago, the percentage of splay legs we see in our experimental farms has decreased from 1% more or less to about 20 to 30%. And the vitality of the pigs, so here I expressed it as percentage, so the observed vitality increased with about 2 to 3 percentage points. So we saw we were making progress within our own farm. And then if we validated these data with the independent data set, we also saw that breeding values, if we calculated them from both data sets independently, were positive, indicating that these breeding values were transferable to other pig breeders, to other farms. So we were effectively selecting pigs for more vitality and decreased incidence of congenital defects. So this is just a nice example of how a really simple phenotypical measurement can be effective in improving vitality, and in that sense also, I think, resilience of pigs at birth. So that was a nice study. And the second study in my PhD, we took it a bit further, a bit more complex. We looked at the genetics of antibiotics usage in pigs. So this study was done a few years ago and also published. I added the link to the article below, and also in collaboration with the Flemish Pietra breeding organization. So here we had two main research questions. First, can we quantify antibiotic usage in pigs? Because it's not a straightforward thing to do. It's not something that is done routinely, I think. And the second, we wanted to look at the genetic component to see, like, can we breed for antibiotic use, and how is it related to other important traits? So for this, we had medicational records, unfortunately not on the individual level, but on the pen level, from the breeding organization. So how the setup of the experimental farm worked was we had different promising Pietra sires, so young sires, of which semen was taken, mated with hybrid sows, offspring generated, and these offspring were kept in different pens with about 6 full sip piglets per pen. So because we had full sip pens, we were able to connect the pedigree, connect genetics, and estimate genetic parameters. And from these pens, we knew which pens were medicated with which antibiotic and what amount. And we also knew the number of pigs that were treated. So this we had these data of about 2,000 pens, where about a third of them were treated at least one time. So at least one pig was treated once with an antibiotic. If we first have a look at the number of treatments over time, we see that, like, this kind of data, it has a huge variability. So you see peaks, so those are probably periods with an infection or something like that, combined with periods where there's even no antibiotic usage. The reason that farm A stops in the middle, because it's just because it was shut down. It's not that it suddenly became very healthy or something. So we had these antibiotic usage data. That was interesting because we wanted to look, like, can we use these data as an improved way of phenotyping pigs for one, antibiotic usage, and second, also their resilience, because antibiotics will only be used if pigs show some kind of sickness or disease or illness or something like that. So whereas we usually have binary records for these kind of traits, like is it pig dead or alive, is it medicated or not medicated, we came up with a new way of looking at it, and we calculated the total amount of active compound of an antibiotic that was administered per kilogram pig of at risk in the pen. So we actually try to quantify it on a continuous scale. We analyzed this, and at the pen level, we had remarkably high heritabilities, 30 to 50%. So I have to say this is at the pen level, so it's a combination of 6 pigs. So that might be a reason why it's inflated a bit. But and if we only looked at respiratory treatments, it's dropped significantly to 1 to 15%, but still it was significantly larger than 0. So we were quite confident that this antibiotic usage has a genetic component. We have also further evidence for this. So a study of Austin Putz a few years ago also showed that the number of medical treatments had like a heritability of 13 to 30%. So this is nice evidence that we can select pigs directly on their antibiotic usage. So it clearly has a genetic component. And then the next question was, what is the relationship with economically important traits? And also there we saw that there was a favorable correlation with mortality, which was expected because pigs that are treated more were probably also more sick or more diseased and have a higher probability of dying. But this indicates or seems to indicate that breeding for lower antibiotic usage would also result into a lower mortality, which is beneficial, of course. And on the other hand, we did not find any adverse genetic correlations with production parameters, so that was also nice to see. So these show that including the trait offers good perspectives for pig breeding for increasing pigs' resilience. But there are some remarks. We only had this study on one parental line, the Belgian Pietrais, and we also only looked at fattening pigs. So in pigs, actually, most antibiotics are used at the suckling or the weaning pigs. So the question is what the genetic correlations are in time. And then also, what would the transferability be to other breeds, and other environments? Plus, we only had data at the pen level, so it would be very interesting to look at this at the individual level. But unfortunately, I haven't been able to look at this during my PhD. But this would be very nice if this could be researched by following researchers somewhere. So I invite you all to do this. So this was a nice paper, but then we took it to the next step in terms of quantifying resilience. We teamed up with Hendrix Genetics, and we tried to quantify resilience as these deviations in body weight, feed intake, and feeding behavior. So this also resulted in a nice paper I've added below. But so here, we really wanted to see, like, with the resilience hypothesis, like, if you have an optimal production level and you deviate from that, how are these deviations heritable and how are they linked to the resilience status of an animal? So to do this, we had a quite nice data set of the Pietrais of Hendrix Genetics. So we had about 6,000 pigs. With a nice pedigree depth and also a lot of genotype information, so we could apply a single-step GBLUB. So here we had this automated feeding station data, and when quantifying resilience with this kind of data, it's always very important to perform rigid quality control, because these machines are not perfect. They are quite noisy sometimes, and they're also prone to technical errors. So if you look at the raw data, you see like you have a lot of body weight measurements of 0 kilograms, for example. That's just not possible, of course. And you also have these inflated weights. So in the first step, it's important to remove these clear outliers. But also in the second step, we looked at pen level deviations, and here you see that's the top left panel A. If you look at the root mean square error of the deviations in weight on the pen level, you see there are a few pens seriously deviating, and probably something went wrong technically there. So what you would expect is panel B, so that's quite nice. Individual pigs are colored with a separate color here. And you see now and then there is an observation outlying, but in general, pigs form a quite normal distribution. But what you also see often is, like in panel C, that at the start of the experiment, that pigs still have to get used to the automated feeding station. They tend to enter it with 2 pigs at a time, causing weights to be doubled or inflated at least. So if you do not correct for these kind of things, the rest of your analysis will be completely bullshit. And then other things you see, like in panel D, is that you get clear technical errors. This is some kind of machine error where the machine resetted its 0 point or something like that, and you had these really strange weight trajectories. So apart from these population quality control, I also performed an individual quality control to even further smooth the curve and only keep data points that were reliable. So here, see, this is a pig that deviated a lot. So probably a challenge will have occurred around 100 days of age and around 130 days of age when the pig lost weight, after which a recovery happened, and then it lost weight again and recovered again. So this will probably have been a challenged pig with a low resilience. If you do proper quality control, you can start quantifying resilience from these data. So we used the following approach. You can see it in the figure on the right. On top is an example of an animal which showed barely any deviations, which had a quite straightforward weight gain over time, whereas the bottom individual was severely challenged, showed quite some deviations. So we used a few different kinds of modeling. First, we used Gompert's growth curve modeling, so those are panels A and D, to predict the optimal weight or the expected theoretical weights of a pig. And then we looked at how much the observed weights deviated from this expected weight. And then you can see in panel B and E that if you put it on the same scale, you can see these deviations can be quite significant for the lower animal, where it barely changes over time for the upper animal. So this should be a quantification of these animals' resilience. And then we had another approach, that's in panel C and F, where we not looked at the individual animal, but we compared the Z-scores. So we compared the animal's weight at a given day with the average population weight. So for example, the top individual at panel C, it has a weight at start which was 1 standard deviation below the average population level. But it fairly remained at that level throughout time. So it was below average, but it was quite constant below average over time. Whereas if you see animal of panel F, so the bottom animal, it started as an average animal, but it decreased soon to an animal with a weight 3 standard deviations below the population mean, after which it recovered, had another drop, and recovered again. So the idea is these deviations can be quantified. We can estimate a variability, a variance from it, and we took the natural logarithm to rescale it, where the idea is that animals with a higher variability, so higher L and VAR, have a lower resilience, and they have shown more deviations from the optimal performance. If we analyze this genetically, we saw that these traits were heritable. They even had quite nice heritability. We also did the same for feed intake. We had heritabilities of like 11 to 23%, which were quite nice. So these show that also these traits are heritable. We can select on them. And it's important to note that if we did not apply any adequate quality control, these estimates will drop below 5%. And that's obvious in our case, because if you do not remove outliers or technical outliers, technical noise, your deviations will be inflated and you will not find any genetic variance, but purely residual variance. And if you looked at genetic coefficients of variation, they were 22 to 30%, which indicated there was a high potential for genetically improving these traits. So very promising. And also here, if you looked at genetic correlations, Within these resilience traits, they were quite high, as expected, but they were definitely not equal to 1. So that means, we believe, that deviations in body weight are not the same as deviations in feed intake, for example. So they share a common genetic basis, but there are still differences among them. And so for breeding values of breeding programs, that would mean that you can add value with regards to resilience phenotyping by taking both into account, I believe. And then furthermore, these traits were beneficially correlated with feed conversion ratio, which is also nice. So more resilient, so less variability, was a better feed conversion ratio. Whereas with average daily gain, it was unfavorable, but it was really lowly correlated. And with muscle depth, was barely any correlation. So those were also very promising. So these results show we can select on them, we can breed for resilience. But then the question is, this is based on a theory. Do we know if this is actually related to animals' health and welfare traits? And to study this, we set up a new experiment where we were looking at the association of these resilience traits with specific health and welfare related traits, such as tail biting, lameness, and mortality. And I've written a paper on that, which is currently under revision, so that should appear soon, I hope. So what was the setup here? So I individually weighed about 2,000. There it is again. I weighed about 2,000 growing pigs in an experimental farm every 2 weeks. So from the start of the finishing phase at about 70 to 80 days of age to about slaughter age, like 180 days, they were weighed every 2 weeks, where we kept only pigs with a minimum of 5 records. In addition to that, we also scored physical abnormalities during weighing. So we had repeat measurements on pigs' tail biting wounds, ear biting wounds, ear swelling or hematomas, and umbilical hernias. So similar to the previous study, we quantified deviations of observed weights versus expected weights using the same techniques, where here I also show on top a pig with barely any deviations from its expected growth curve. So panel A, B, C, whereas the bottom pig, it starts out quite nice, then it comes across a serious challenge, loses a lot of weight, and deviates a lot. So here again, a higher LN-VAR means a lower resilience and more deviations from an optimal performance. If you looked at heritabilities here, they were even higher than in the previous study. That might have been due because the previous study was in a high health farm of Hendrix Genetics in a purebred line, whereas this was in commercial conditions in crossbred pigs. So this might, that's my theory, might cause more challenges to happen within the stable environment and to be able to express their resilience better. Because if there are no challenges, you can't express any resilience because you're not challenged by anything. But so they were quite heritable. So that was again nice, and it offers good prospects for selection. If we then also looked at the scores for tail biting wounds and ear biting wounds, we were also there we saw they were heritable and also quite heritable, so lowly heritability for tail wound scores and a higher heritability for ear wound scores. But there we also saw there was a very large pan effect, which is logic in a way, because if these pigs, they were kept together in pans of full sip and half set pigs. So if there's an event of tail or ear biting occurred, it quickly spreads to affect the whole pan because it's some kind of social trait. Furthermore, we saw for lameness, umbilical hernias, and mortality that these were lowly heritable, probably partly due because these are, again, binary records with a low prevalence, causing there to be little variation, and yeah, that's not ideal for a breeding program. So the idea here is maybe we can just select on the variability in weights and exploit that as a correlated response to simultaneously improve the other traits as a correlated effect. If we just looked at phenotypical associations, we did saw a really significant association between these deviations in body weight and mortality, lameness, and tail biting wound scores, whereas we did not find this for ear biting wound scores, hematoma scores, and umbilical hernia scores. So this was it was nice to see that was correlated with the top ones. We have a theory why it was not linked with ear biting scores, for example. We saw that ear biting wounds mostly appeared or were visible upon arrival at the farm. So pigs usually already had bitten ears when they arrived in the farm. And so this means, or that's what I believe, that the challenge of ear biting will probably have occurred at the stage before the finishing phase. And so the deviation will probably already have occurred earlier. And that's why I think these deviations in body weight were not present in our phase, the finishing phase, because there they mainly recovered. For hematomas, yeah, that's also something that usually appears very early in life. Very often when pigs are getting ear tags, so then they get an ear swelling. So also there, the challenge might have happened long before the finishing phase, and that might be a reason why we don't see an association with this Allen-Varg parameter. And then for umbilical hernias, here we think that this is more chronic. So umbilical hernia is an expulsion of the umbilical region. So we don't think this is an acute disease, and maybe that caused pigs not to be deviating a lot in body weight. But what was interesting, if we looked at specific bands, where, for example, tail biting events appeared, we clearly saw that, so here you see all these pigs, if they have no visible wounds on their tail, they're colored red. From the moment they start coloring green and even worse blue, their tails have been bitten, and you see that this has a serious impact on their weight for some pigs, while others are marginally affected and recover afterwards. So this clearly shows the correlation between tail biting and these deviations in body weight. So as said before, next to these phenotypic correlations, we also looked at genetic correlations, and here we also found a really significant genetic correlations between these Allen-Varg parameters and tail wounds, tail biting wounds, lameness, and mortality. Next to this study, there is also other evidence for this relationship. So between these resilience traits based on deviations and resilience indicators. One nice study is this of Austin Putz again, where we looked at a resilient trait based on daily feed intake deviations, also in crossbred pigs and a natural disease challenge. And there we saw a really positive genetic correlation between these traits and mortality and the number of treatments. So these observations suggest that by looking at longitudinal data, by quantifying deviations, we are really able to quantify resilience, and this should allow us to breed for more resilient pigs we really need for future pig farming. So I'm concluding my presentation now. What I want you to take home, there is a growing need for resilient pigs, and pig breeders desire new resilience traits to effectively achieve this. We have been able to develop new promising resilience traits. So based on weight and feed intake deviations, they have low to moderate heritability and favorable correlations in general. And this might offer a practical way to really increase pigs' overall or general resilience. Where I have to say that future research still has to look at the relationship between purebred and crossbred performance, because it's not clear at this point how translatable these breeding values are from a purebred line, for example, to their crossbred offspring, mainly because the farm environment can differ significantly between both systems. But with this, I thank you all for your attention, and I am happy to answer any questions.
Thank you so much, Wim. Appreciate it. Very large study you've done here. First of all, I want to congratulate you on putting the emphasis on the quality control. I hope there are many students here today listening. Don't look at the data before you analyze them. You know, look at the outlier, especially when you look at this type of traits, because things can change, and then the results change so much at the end you've not been. So I really appreciate that, and it shows quite well how that was beneficial to your study. So I'm going to take a couple of questions from the chat box. You can stop sharing if you want. Wim?
Yeah.
And you can read it. The first question from what trend is coming next in pig breeding after the three you indicated?
What trend is coming next? Vaccines. Look, can we just really stick back? Yeah, I know Peter Bijema is really busy with trying to eradicate diseases. So breeding to really not reduce disease incidence, but eradicating specific bacteria, for example. That might in pig production also be really useful, or an opportunity with the PRS. But at this point, I think increasing resilience and then maybe also social behavior will be the main traits for the next decade, I think. Because certainly in Europe, there's a growing tendency for going towards cage-free systems. So sows that need to be able to deliver their pigs outside of a closed pen. Pigs that need to be able to go outside. So in those systems, you need good animal behavior. So in first instance, good mothering behavior, but also good social behavior, where your pigs also are friendly towards each other.
And there is the next question from Plush. Will the variability in antibiotic usage, different antibiotics used, affect the genetic component of it?
Yes, I think it definitely will. So we had we were lucky. We had a quite standardized data set from 2 farms where they used the same antibiotics, and they had fairly the same scheme of treating pigs. But here, that's another danger for transferability. I don't know how well this would correlate, these breeding values, with another farm where they use other antibiotics with another protocol, let's say. So that's something I really believe there is a core genetic component to it, but at this point I cannot say to which extent it will be affected.
And then the last one from Yorgos, which I think you touched, but about the heritability of antibiotic usage, do you think it's also mostly due to maternal?
So maternal effects. So in our study, we looked at it from a Pietra's side, so a paternal sire line. So the genetics we estimated were mostly due to paternal genetics. Although I think, no, I used an animal model. But we could look at that by using a sire model, because if you would take a sire model, you would leave out the maternal genetics. But I think, it's been 3 years since I did the study. But I think maternal genetics was involved, but still the sire genetics had a really large involvement as well. So there was a really pure genetic component to it, and not just maternal effects, let's say.
Thank you. We have 5 more minutes for even general discussion, for any other, also for Gregor and Gábor. Gábor, do you have a question for Wim by chance?
In fact, I do. No, I just was when you were talking about this, so that you could apply just for pen basis and not individual basis. I would just ask for your opinion on what would you expect, that what would happen if you would be using it in an individual basis, and for example in different species like perhaps cattle or chicken.
So I think individual data, I think it would allow us to increase the accuracy of selection, because now heritabilities are inflated, but within a breeding program, if you look at the pen levels, even though your heritabilities might be a little bit higher, you only have 1 or 2 observations. So I think by that simple fact, your accuracy will go down and your genetic progress will also be lower. By transferability to cattle, for example, that's something I'm really interested in, and I don't know if there are any papers on that, but I think especially in the Nordic countries, they have a tendency of really keeping track of medicational records. So I think the data is there, but I haven't seen a paper yet on if they really looked at the antibiotic usage as I did. But I would assume it would have a lower heritability because you have a lot more environmental variation. Because cows are usually not kept in these highly standardized intensive farming systems. And for chickens, there will probably be some genetic background, but chickens are usually vaccinated at birth or in large groups. So it's really hard to detect any variability between different chicks, I think.
Yeah, thanks.
All right. I guess at this point, if there are no other questions, I really would love to thank 3 fabulous speakers. Guys, you've been great. Really appreciate. I want to thank Neogen for sponsoring this great webinar, and the audience, which came numerously, which we always appreciate, and give good questions as well. So thank you so much, and thank you for Federico at the EAAP Secretariat as well for the support and all this team.